Hello students, welcome to my class. I am Dr. Sujata Chaudhuri, Head and Assistant Professor of Department of Zoology, Barakpur Rashtraguru Surendranath College. Today I will discuss with you a very common topic, that is plasma membrane. Now, I have referred plasma membrane as biomembranes. So, why so? The biomembranes are the basic membranous structure of any kind of cells, be it animal cell or plant cell, and also it is outer boundary of any type of cell organelles like mitochondria, Golgi bodies, endoplasmic reticulum. Whereas the name plasma membrane or cell membrane is referred to the only outer boundary of a cell. So, all type of cellular boundaries or the boundaries of cell organelles are referred together as biomembranes. I have divided this topic into two subtopics. First one is the structure. But before going into the details of this topic, I am referring here the structure or glycerol. So, glycerol is a 3-carbon molecule with 3 hydroxyl group. Now, upon esterification, these 3 hydroxyl group can form 3 hydrocarbon chain and then it is called triglycerides. You can see here the glycerol molecule is attached to 3 hydrocarbon chain. Now, if the hydrocarbon chain is so that each and every carbon atoms are attached with the adjacent atoms or carbon atoms in a single bonded condition, it is called saturated lipids or fats. Whereas, if the carbon atom is bonded with another carbon atom by double bond or triple bond, it is called unsaturated fatty acid or lipids. In the upper portion, you can see that the tail or the hydrocarbon chain is just straight because all the carbon atoms are lined in a single bonded condition, whereas in the lower structure, the carbon where is bonded with another carbon with double bonds, it is showing a bend or kinks. So, this is the structural uniqueness of unsaturated fatty acid. Now, coming to structure of a phospholipid. In the previous case, we have found that the 3-carbon glycerine molecule is forming 3 chains. But in case of phospholipid, the 2 carbons are forming 2 fatty acid chain or fatty acyl chain. One is straight, that is saturated chain, and another is bent one, that is unsaturated chain. And the 3rd carbon here is not forming any chain or tail, but it is bound to a phosphate ion. Now, we know that phosphate ion has negative charge. So, because of this condition, the head region or the upper domain of this phospholipid is hydrophilic in nature. So, the hydro means water and philia means loving. So, the upper portion or the head region of a phospholipid is polar in nature and has affection towards water, or you can say that they can freely stay inside a hydro environment. Whereas, the lower portion of the lower domain of the phospholipid is forming by the hydrocarbon chains. That is why they are hydrophobic in nature. So, phobia means fearing, hydro means water. So, the tail region is water fearing domain of a phospholipid. That is why the phospholipids or this kind of lipid molecules are showing a unique characteristics that is called amphipathy. So, these molecules, this kind of molecules have a particular domain that is hydrophilic and another domain that is hydrophobic. Now, if this kind of lipids are put into the water environment or water medium, what will happen? All the hydrophilic head groups orient themselves in such a manner that the head region faces the water and all the hydrophobic tails gets buried inside this structure so that they do not come in contact with the water medium. This kind of structure is called micelle. So, we have seen that if we put oil into water, this kind of bubbles will form where we can see that the hydrophilic head regions are orienting themselves towards the water medium, where hydrophilic regions or the tail regions get buried inside the bubble. Now, if the condition is such that both the outside and inside of this structure has water, so what will happen then? The lipid will form a bilayer or a bilayered wall where the hydrophilic heads will face both the outside and the inside structure, and in between them the hydrophobic tails regions will get buried. So, this kind of structure is called liposome. In the lower structure you can see that it is forming a bilayer sheet where the upper and the lower layer is formed by the hydrophilic heads, and in between them the hydrophilic tail regions are secured from the water. During the origin of cell, we think that these kind of liposome structures were created to form a double layered boundary where the cellular reactions and their products were secured from the outside chaos of the primitive soup. So, maybe the origin of plasma membrane is the first sign of origin of a cell, where cell can get its integrity apart from the outer world. Now, coming to the history. After discovery of microscope, we all know that Robert Hooke first showed that all type of plant and animal tissues are made up of cells. Here we can say these are the cork tissue made up of plant cell, and we can see that the plant cell wall was easily visible under microscope, but no such cell boundary was found in case of animal cells. So, the nature of the animal cell membrane was first correctly depicted by Quincke, who noted that if we cut a cell and put it into the water, it will form a bubble or spherical shape. If we again cut that small sphere and put it into water, it will again form smaller spheres. This kind of behavior is only exhibited by oil or lipid. So, somewhat he correlated that the cell boundary must be made up of oil or lipid molecules. After that, E. Overton in 1890 showed that membranes that proved that it has a facilitated diffusion through the lipid membranes, and membranes may be made up of fatty oil or oil. The groundbreaking work from Gotter and Grendel was that they extracted the lipid from RBC or red blood corpuscles. So, this is called red ghost. Why? Because RBC lack nucleus and many cell organelle. So, if we can lyse the cell or RBC, the most amount of boundary structure or the components can be extracted. They then calculated the ratio of surface area of these extracted lipids with water divided by the surface area of RBC as a whole or intact, and found that this ratio is 2:1. So, first they concluded that the layer that surrounds this RBC or any cell is double in amount. So, it is not a monolayer lipid, but it is a lipid bilayer that surrounds a cell. But Gotter and Grendel experiments show that the surface tension of RBC is somewhat different if the total surface is made up of lipids. So, they depicted that maybe there are some other components. Dafson and Danieli in 1935 proposed that along with the lipid bilayer, there is a layer of protein on both the inner and the outer surface of a membrane. So, they proposed the model of sandwich, where the inner core of a membrane is made up of lipid bilayer and the outer and the inner layer is made up of a layer of proteins. So, it is just like a sandwich where protein, then lipid bilayer and then protein forms the total membrane structure. In 1972, Singer and Nicolson first proposed their model of membrane, that is Fluid-Mosaic model, and till now it is most accepted model of plasma membrane or any type of biomembrane. This model shows that lipid bilayer of the membrane are in fluidic state and form the backbone of biomembrane, where this lipid molecule can move laterally and freely within the plane of membrane, and within these lipids there are patches or mosaic or discontinuous particles of protein that penetrates this lipid bilayer. So, how this fluidity is maintained of a membrane? The lipid and the mosaic protein patches or the molecules can move from one place to another within this membrane, either laterally, that is sidewise, or they can rotate through their own axis, or they can jump from one leaflet or one monolayer of plasma membrane to the another monolayer. So, vertically they can jump or flip flop from one leaflet to another leaflet, and this movement is mediated by an enzyme that is called flippase. Now, going into the details. It is the phospholipid structure. The lipids that contains a phosphate group is called phospholipids, and the membrane lipids are usually derived from glycerol backbone, that is why they are called phosphoglycerides. The phosphoglycerides here in case of membrane is diglyceride, as they have only 2 hydrocarbon chains or hydrophobic fatty acyl chains. That is why they are called diglycerides, and the third carbon, as I have already told you, is attached to a phosphate. So, they actually form an perfect amphipathic molecule having a hydrophobic tail region and hydrophilic head region. Now, this phosphate group can be attached with other reactive group. Here in this picture you can see that there is choline. Now, depending upon the reactive group that are attached with the phosphate, the phospholipids can be of 4 types. If it is attached with ethanolamine group, it is called phosphatidylethanolamine. If it is attached with serine, it is called phosphatidylserine. If attached with choline, it is called phosphatidylcholine, or if attached with inositol, it will be called as phosphatidylinositol. Apart from these phospholipids, there are some other lipid molecules that are called sphingolipids. Now, and the last structure, you can see that this is the structure of sphingomyelin. Another very important component of any membrane is cholesterol. So, it is not a typical lipid molecule, but it forms about 50% of membrane lipids, but it is absent in plant or bacterial cells. In plant or bacterial cells, these cholesterols get replaced with sterols. You can see the structure of cholesterol. It is has some kind of aromatic rings. It is a flat and rigid structure. So, abundance of cholesterol interferes with the movement of fatty acids and protein molecules. So, if there are many cholesterols, the movement of the membrane will be interfered and the membranous structure will be more restricted. Now, the both leaflet of a plasma membrane means the extracellular leaflet and the cytosolic leaflet is somewhat different depending upon their composition. So, outer leaflet of a plasma membrane is mainly formed by phosphatidylcholine, sphingomyelin and glycolipids, where the cytosolic part of the monolayer contains more of phosphatidylethanolamine, phosphatidylserine and phosphatidylinositol. Cholesterol is distributed in the both leaflet in a 50% manner. Now, another important component of Fluid-Mosaic model is the proteins. So, proteins in the membrane are of three kinds. One is called integral membrane protein. It is also called transmembrane protein because this kind of protein span the whole membrane from outside to inside. So, they have two different domains. One is hydrophilic domain that faces the cytosolic part or the extracellular watery part, and a hydrophobic domain that gets bonded with the lipid bilayer or the lipid tails. So, the examples of these integral membrane proteins are ion channels, proton pumps. You can see that the ion channels or the proton pumps is actually function as a carrier protein or the pumping system to get exchange or in and out movement of the ions in and out of a cell. Second type of protein is called peripheral membrane protein. These peripheral membrane proteins is attached to the surface either on the extracellular part or in the cytosolic part, but it does not traverse or span the whole membrane and it can get discharged in frequent manner. So, some enzymes are of this kind. The third proteins is lipid anchored proteins that gets covalently bound to the lipid molecules. So, G protein, a very important protein in signal transduction, is lipid anchored protein. The third component of a membrane is carbohydrates or sugar. Though it this composition is around 2 to 10% of total composition wise, but it performs very important part in cell signaling and cell-cell interaction. And one thing, the sugar moieties always face the extracellular part. It is never present in the cytosolic part. This sugar, if attached with the protein molecule, is called glycoprotein. If attached with the lipid molecule, is called glycolipid. to summarize and add some points into the structure of biomembrane is that the entire membrane width is 60 angstrom, 30 for tail region and 30 for the head region of lipid bilayer. So, lipid bilayer provides a structural and fluidic backbone of membrane and provides a barrier so that any kind of random movement is not occurring in and out of a cell. The protein that are present in a mosaic patches carry out specialized function such as transport across the membrane. Again, another thing very important is that the protein and the lipid ratio varies from cell to cell depending upon the function of the cell. The protein is very high in mitochondrial membrane as mitochondria is engaged in cellular respiration and needs many proton pumps and another type of channel proteins. So, the protein part in mitochondrial membrane is very high. It is rather less in RBC and it is very less in myelin sheath where lipid portion or the lipid is very high in myelin sheath, as we know that nerve cells or the nerves gets bound with the myelin so that it can act as a electrical insulator and electrical propagation can be held in a smooth manner without any leakage. And the last point is the carbohydrates play important function in cell signaling and interaction. Now coming to the second part of this lecture, that is function of biomembranes. Now biomembranes, you can say that it is the largest cell organelles with many functions. To name some of them is compartmentalization. So plasma membrane form an unbroken and continuous barrier that safeguard the inner content inside and allow specialized activity to proceed independent and without any external interference. Second, it is forming an intracellular scaffold. So, membranes provide the cell with an extensive framework or scaffolding to maintain its shape and the transport of cell organelles. Third is selective permeable barrier. So, that means they provide a means of communication in and out of a cell by promoting selective transport across the membrane, because cell is a live system. It needs energy, it needs many solutes to come inside the cell, it has to expel some excretory material outside the cell. So, it is not a closed system, but an open system. So, some kind of transport must be present in cell and that is done by the membrane structure. The membrane is the outer surface of any cell, so it responds to external signals. So, membrane has various type of protein receptors that can recognize and bind to specific molecules that are called ligands and act accordingly. Then intercellular interaction. The cell is not a alone creature, it has neighbors and it needs to communicate with its neighbor to facilitate recognition, cell-cell adhesion, exchange of materials and information. The last point here is energy transduction. We know that energy conversion is a very important part of any living system. So, the membrane or the biomembrane of plastid, we know that is involved in photosynthesis in plant cell or in mitochondrial membrane that are engaged into electron transport chain forming the ATP. I am focusing only to one part of function, that is transport across the membrane. Now, this mechanism can be of two types. One is passive transport another is active transport. The passive transport means this kind of transport mechanism does not involve any expense of ATP. So, ATP is the money, you can say it is a cellular currency that are not used in case of passive transport. The molecules or reagent can diffuse in and out of a cell or with the help of another protein, but there is no expense of ATP. In opposite case, the active transport needs the expense of energy, that in form of ATP, and usually it occurs against the concentration gradient. So, from lower to higher, it is the against the concentration gradient. The first part, that is passive transport, involves three kind of mechanism. The first one is diffusion. So, if we put two different type of molecules in two different compartments and allow them to transport in between freely, so what will happen? The reagents will mix with each other freely until they reach to an equilibrium, where in both the compartment each of the molecules are equal in concentration. So, the diffusion is the movement of ions or molecules from region higher to lower concentration, that is in favor of a concentration gradient. The molecule that pass by mechanism of diffusion depends upon the polarity, size and charge. The molecule that can pass easily through this lipid bilayer is hydrophobic molecules, that is water soluble, oil soluble, that is oxygen or nitrogen gas. These can be non-polar molecule like benzene, or small uncharged polar molecules like urea, glycerol or carbon dioxide that can pass freely through the lipid bilayer. Molecules that will not pass this lipid bilayer are large uncharged molecule like glucose, polar molecules like sucrose, ions like proton or sodium ion. So, any kind of charged molecule will not pass through lipid bilayer, but a cell needs these molecules to get entry into the cell. Here comes the effect of protein molecules. Another type of passive transport is osmosis. So, osmosis is the movement of water, only the water or the solvent from a region of higher concentration to a region to lower concentration, and in between there will be a semi permeable membrane like cell membrane or plasma membrane. So, in this condition, in the middle picture you can see if the isotonic solution, that is the salt concentration of this solution is equal to the salt concentration of a cell. So, if cells are put into this isotonic solution, what will happen? The water molecule can freely get into the cell and get outside of the cell, but the total overall shape of the cell will not fluctuate. In hypotonic solution, the first slide or the first picture where the salt concentration of surrounding medium or outside medium is low. So, hypo means lower concentration. So, what will happen? In order to make an equilibrium, the water will come from outside into the cell and the cell will swell. If it swells so much that it will burst upon, and this is called lysis. In case of plant cell, the cell wall will not allow such lysis, but the cell will be very swollen. This condition is called target solution. Okay. The last stage you can see that the hypertonic solution, where the salt concentration outside of a cell is very high. So, what will happen? The water from cell will move out from the cell and the cells will shrink. This condition in plant is called plasmolysis. The third type of diffusion mechanism is facilitated diffusion. So, here the diffusion occurs with the help of a protein molecule that is called permease. This time the rate of diffusion gets accelerated by some membrane protein that act as a carrier molecule and help in this diffusion. So, the rate gets double or many. Next, another type of transport mechanism that need ATP as a energy expenditure is active transport. The active transport requires energy, as I have already told you, in form of ATP. It is mediated by membrane proteins. It pumps materials across the membrane against the concentration gradient, that is from low concentration to high concentration, and it is of two types. One is protein channels, another is carrier proteins. So, what are protein channels? Protein channels are the protein molecules that permit transport of molecule across the transmembrane protein having a central tunnel or channel. It can be simple channels just with the aqueous center that permits ion or another things to diffuse in and out of a cell, or it can be gated, means in most of the cases it will be closed, but the gate will open only by either voltage or ligand or some mechanical means. Means voltage gated channels will open only by some change in ionic charge. Example is potassium channels. The ligand gated channels will open by binding of a specific type of ligands like hormones or antibodies, like in calcium channels it is CAMP by cyclic AMP. In mechanogated channels it needs some mechanical forces to open the gate, like respond to sound, the internal ear cation channels will be opened. The second type of protein that help in transport are the carrier proteins. So, carrier proteins permit transport by binding to the molecule to be transported, and then by undergoing a structural changes, that is called conformational changes, to release the molecule to its destination. It can be of three types. Uniporter means it transport only one substance, that is in case of glucose pump, the glucose can be get entry into a cell. Symporter means two substances at a similar time will be transported in the same direction, like sodium glucose pump in intestinal cells. So, sodium and glucose will be pumped inside this kind of cells simultaneously in a same direction. Antiporter transport two substances simultaneously but in opposite direction, like sodium potassium ATPase pump, where the sodium gets outside and potassium get transported inside in expense of one molecule of ATP. Here are the three type of structural difference of carrier proteins. First one is uniport, where one single substance is imported through it. The second is symport, where two kinds of substances are getting transported, and antiport, one is going inside, another is going outside of the cell. Now, this portion is very important and bit complicated also, that is sodium potassium ATPase pump. So, we can see that the first point means where the pump protein is opened in the cytosolic part. Now, in this opened condition, 3 sodium ion gets bound to this protein upon phosphorylation of a ATP molecule. Now, you can see that ATP is converted into ADP and the phosphate ion gets attached, or it is phosphorylating the pump system. Now, upon that phosphorylation, the palm protein will get some conformational change, means its structure will be changed and it will be open to the outside. So, upon opening, the sodium ions will be released into the outside environment. Now, in this condition, when the palm protein is open to the outside environment, the potassium, 2 potassium ion will be bound to this palm. Upon this binding, the palm protein will be dephosphorylated, means the phosphate gets detached from the palm. Upon this detachment, the palm protein will get another conformational change and it will be like the original state, that is now it is open to the cytosolic part. So, the potassium will be released into the cytoplasm. Again it will be binding to 3 sodium ion and this cycle will be goes on. So, in exchange of 3 sodium ions, 2 potassium ions will be entered into the cytoplasm or within the cell, and for this whole process 1 molecule of ATP will be used. Now, up to this level we have talked about by the molecular transport system. If cell needs some bulk transport, okay, so what will be happening? This bulk transport is of 2 types. One is endocytosis, means when the large amount of materials needed to be get inside the cell. So, that is called endocytosis, and when large amount of secretion or excretory products need to be expelled from the cell, it is called exocytosis, that is outside of the cell. Endocytosis is of 3 types, that is we know that phagocytosis or cell eating, pinocytosis or cell drinking, and the third one is receptor mediated endocytosis. Now, in case of phagocytosis, the molecules that are needed to be bulk transported first gets contact with the plasma membrane. So, the plasma membrane makes an invaginated vesicle, and afterwards the invaginated membrane detaches to form small vacuoles or vesicles, that is called phagosome. In case of pinocytosis, the same process is repeated, but instead of solid material, here fluid is taken inside the cell and the vacuole is called pinosome. The receptor mediated endocytosis is basically uses different kinds of receptors. So, receptors are present on the outside surface of a plasma membrane, binding with a specific type of ligand, that is hormone or antibody, to the receptor proteins, form a receptor ligand complex. Then this ligand receptor complex move laterally towards a coated pit. We know that the protein molecules and the lipid molecules can move freely, that is why it is called fluidic or the Fluid-Mosaic system. It can move laterally to a coated pit. So, pit is like well like structure. So, many of these ligand and receptor complexes gather there, and this pit consists of a protein that is called clathrin. So, cytosolic part of this pit contains clathrin, and clathrin is a 3-armed protein that forms a cage around this pit with other type of clathrin protein and form a coated vesicle. These coated vesicle then pinch off from the plasma membrane and go deep inside the cell, where this clathrin protein sheds off and form a simple endosome. Then the ligands are released from the receptor and the vesicle is now called curl or CURL, that is compartment of uncoupled receptor and ligand. So, a single vesicle will be divided into two vesicles. In one part there will be receptor proteins and another part will be carrying the released ligands. Now, the receptor part of this vesicle will be detached from the ligand part and it will be recycled back to the plasma membrane, so that it can catches more such kind of ligands again, and the ligand part, that is in the endosome, will be processed further. To summarize the whole part of the function of plasma membrane, we can say that plasma membrane or any kind of biomembrane is the largest cell organelles with many functions. The selective transport across membrane help to survive the cell and function individually among the chaos of surrounding world. Protein molecules carry out very specialized transport system, that is via passive or active mechanism, and if cell needed some bulk transport, it can employ different mechanism that differs from a molecular transport. So, these are my references and thank you all.